So good afternoon, everyone. It's Ivan Campos here. Thank you for joining us in our first CGIL seminar of the year. Today, we are happy to have with us Shannon Cartwright. So Shannon is currently a PhD candidate in the Department of Pathobiology under the supervision of Dr. Bonnie Mallard. She graduated with a Bachelor of Science in Agriculture from the University of Guelph in 2007. She then promptly started a master's degree in bovine immunology under the direction of Dr. Bonnie Mallard. The focus of her thesis was to compare the immune response and survival between purebred and crossbred cattle. She successfully defended her master's in 2010 and then began working as a research assistant and was involved in a project that focused on bringing the high immune response technology to the marketplace. In 2011, she accepted a position as a herd manager of a 120 milking cow robotic farm, and she worked there from 2011 to 2013. In May 2013, she returned to work for Dr. Mallard, managing a project that involved developing a genomics test for immune response in dairy cattle. Over the years, she has been an author of 10 peer-reviewed publications, with 4 of these being first author, publishing in journals including BMC Veterinary Research and Scientific Reports. In September 2017, she began her PhD research under the supervision of Dr. Bonnie Mallard. Her current project involves looking at the effects of climate change on the resilience of dairy cattle that have been immune response phenotyped. And so with that, I'll pass it over to you, Shannon. Thank you.
All right. Thank you, Ivan, for that introduction. And thanks for asking me to come and talk to you guys. So as Ivan mentioned, I'm going to talk to you about some of the results we've found so far in my PhD project, looking at the impact of climate variability on the resilience of dairy cattle classified as high, average, or low immune responders. Oh, now I'm not able to change slides. Why is that not working? Okay, there we go. Sorry about that. So this is an outline of what I'm going to go over today. So I'll start with talking about some background for this project, and then state our overall hypothesis and objective. And then I'm going to go on to talk about some of the individual studies that we've completed in this project so far, and then finish off by giving some overall conclusions. So the climate is constantly changing due to increases in greenhouse gas emissions, and this leads to a number of environmental problems, including overall warming of the atmosphere, extreme temperature swings, and increased extreme weather events. And this figure here shows a number of the environmental issues that are associated with climate change. And that includes things like changing in rain and snow patterns, as well as stronger storms. So you might see an increase in precipitation, or in certain cases too, you can see decreases in precipitation. We're also seeing changes in animal migration and life cycles. So because of warmer climates, we might be seeing animals staying in areas longer when typically they would migrate to southern climates, or we're also seeing animals that would not necessarily survive in colder climates. They're now surviving longer. Additionally, we're seeing less snow and ice, as well as thawing permafrost, and that would be in your Arctic regions. We're also seeing more droughts and wildfires, and this was pretty evident in our western provinces this past summer. And then where our research is specifically focusing is we're seeing a lot more higher temperatures and more heat waves. So because of these higher temperatures and increased frequency of heat waves, this leads to a greater risk of heat stress for our dairy cattle. And studies have shown that heat stress in dairy cattle leads to increased mortality and disease incidence, as well as reduced production and reproduction. So heat stress in dairy cattle is typically influenced both by temperature and humidity. So therefore, studies will use what is called the temperature humidity index in order to indicate the environmental conditions at which heat stress occurs. So this formula here is one way that some researchers calculate temperature humidity index, and is the way that we've used to calculate temperature humidity index in the studies that we've done. So some studies have shown that at a temperature humidity index of 72 to 74 is where you'll see physiological changes occurring in dairy cattle. But it's also been shown that for high producing cattle, you can see changes occurring at a THI as low as 68. So there's numerous changes that occur in dairy cattle during heat stress. And initially, you're going to see an increase in respiration rate and sweating. So these are the evaporative cooling mechanisms that dairy cattle will use to try and dissipate heat from their body. Additionally, internally, dairy cattle will start to increase production in what is called heat shock proteins. And these are molecules that are really important for protection and repair of cells. So during stressful events or as body temperature increases, proteins within the cells will actually start to denature. So these molecules are really important for repairing those denatured proteins or protecting themselves from the denaturing of the proteins occurring in general. We also will see that dairy cattle will decrease their feed consumption in order to reduce metabolic heat load, and this can lead to reduced rumination activity, as well as can possibly lead to reduction in milk production and various milk components. Studies have also shown decreases in reproduction, and this includes things like impaired estrus, impaired oocyte development, as well as cattle that experience heat stress late in gestation can have an increase in their circulating prolactin. So their normally prolactin would be transferred into the mammary gland in order to induce milk production and the subsequent lactation. So if they experience heat stress during late gestation, studies have shown that that prolactin doesn't actually transfer into the mammary gland as readily. We'll also see decreases in health and immune function, and this includes things like decreased lymphocyte proliferation, an increase in reactive oxygen species synthesis, as well as a switch to a type 2 immune response, so leading them more susceptible to viral infections, and downregulation of genes that encode complement factors, which is part of the innate immune system. And then at a certain point, if dairy cattle are no longer able to dissipate heat from their body, you'll see an increase in rectal temperature and body temperature beyond normal range. And this would be then considered that the cattle are in a state of hyperthermia. So due to all the issues associated with heat stress, there is a need to identify animals that may be more tolerant to heat stress. So studies in dairy cattle have shown increased expression of genes for heat shock protein 70 production, nitric oxide synthesis, and immune response that are actually associated with thermotolerance. As well as a number of studies from the Millard group have shown the ability to rank dairy cattle as high, average, and low immune responders based on their estimating breeding values for immune responsiveness. And these cattle that are classified as high immune responders have an overall lower incidence of disease and improved response to vaccine and colostrum quality. So therefore, the overall hypothesis for this project was that cattle classified as high immune responders will be more resilient to heat stress as a result of climate change when compared to average and low, with our objective being to determine any differences in resilience in terms of physiological mechanisms and function of blood mononuclear cells of immune response phenotype cattle when exposed to heat stress as a result of climate change. So I'm now going to move on to talking about some of the individual objectives that we completed in this project, with the first one being to look at the effect of in vitro heat stress challenge on the function of blood mononuclear cells from dairy cattle ranked as high, average, and low immune responders. And this study is actually currently published in BMC Veterinary Research. So our hypothesis for this study was that cattle classified as high immune responders will have greater heat shock protein 70 concentration, nitric oxide production, and cell proliferation compared to average and low responders. So for this study, we collected whole blood at a thermoneutral THI from 15 high, 15 average, and 15 low lactating dairy cows that were housed at the Elora Research Station. And then we isolated blood mononuclear cells from the blood and exposed them to 3 different heat treatments. So that included a thermoneutral heat treatment where the cells remained at 37 degrees for the duration of the study, a heat stress 1 treatment where the cells were exposed to 1 heat treatment of 42 degrees for 4 hours, and then a heat stress 2 treatment where the cells were exposed to 2 heat treatments on subsequent days at 42 degrees for 4 hours. And then we looked at nitric oxide production, cell proliferation, and heat shock protein 70 concentration in these cells. And then use general linear models to account for any effects and differences between the phenotypes.
Okay, I think we have problems with Shannon's connection, so just ask her patience, so we'll figure it out how to go back to the presentation. All right, Shannon.
Hi, sorry.
You're back. That's okay.
Yeah, I lost connection. I don't know what got missed and what didn't.
Yeah, I think you stopped in the previous slide, at least for me.
For
but you
the
had
materials
already
and
showed.
methods?
Yeah, but everything was already shown.
Okay.
So
So you
maybe it was
can
the
you see my slides
transition
right
to the
now?
next. Yes, I can.
Okay. So we went through that last slide and then it cut out at this slide?
Yes.
Okay. All right. Sorry about that.
No
Hopefully
problem.
that doesn't happen again. Anyway, as I was saying, so this slide shows an image of our publication that's in BMC Veterinary Research for this study. So if you want to have a look at that, you can. So now I'm going to move on to the results from this study. So this graph here shows our results for HSP70 concentration in the cells. So as I mentioned in the background, HSP70 is important for protection and repairing cells during heat stress. So as you can see from this graph, we found that after our first heat stress treatment, the high immune responders produced significantly greater heat shock protein 70 compared to our average responders, and additionally, our low responders did as well. And then when we got out to after our 2 subsequent heat treatments, we found again that our higher immune responders had a trend towards greater HSP70 production compared to our low responders. And then when we look at within phenotype across the treatments for HSP70 concentration, we found that our high immune responders had significantly greater HSP70 production compared to thermoneutral, as well as had greater HSP70 concentration when we got out to 18 hours after our heat stress 2 treatment. Whereas for our averages, we didn't see an increase in heat shock protein 70 concentration until we got out to 18 hours after our heat stress 2 treatment. And then for the lows, we saw a significant increase from thermoneutral after our first heat stress treatment, but then we did see a significant decrease after our 2 subsequent heat stress treatments. And then when we look at results for nitric oxide production, so nitric oxide is a molecule that is important for defense against pathogens, but studies have also shown that it's a molecule that's important for vasodilation of the skin during heat stress as well. So from our results for nitric oxide, we didn't see really any significant differences except for these thermoneutral treatments. So within each treatment, besides thermoneutral, we didn't see significant differences between the phenotypes. However, when we looked across all the treatments, we did find that there was a significant overall trend for the highs to have increased nitric oxide production when compared to the averages and lows. So could possibly indicate that they have greater vasodilation in their skin during these heat stress events. And then looking at results for cell proliferation. So cell proliferation is really important for defense against pathogens. It's what initiates an immune response to the pathogen. So our results for cell proliferation, after our 2 heat stress treatments, we see that after the first heat stress treatment, our high immune responders had significantly greater cell proliferation compared to average and low, as well as after our second heat stress treatment as well. So might indicate that they are able to better defend against invading pathogens and resist disease during heat stress compared to average and low responders. And then when we look at this broken out within the phenotypes across the treatments, we found that our high immune responders had significantly greater cell proliferation compared to thermoneutral after both the heat stress treatments. We didn't see any significant difference across the treatments for our average responders, and we actually saw a significant decrease in cell proliferation, so might indicate that the cells are actually dying compared to thermoneutral to heat stress 1 and then heat stress 1 to heat stress 2 for our low responders. So I also just wanted to mention this other study that was done by Dr. Ali Livenois in conjunction with the study that I was just talking about. So this study's published in Scientific Reports. And for this study, Ali actually used the same cells that I just was mentioning that went through the different heat stress treatments. And she isolated the DNA from those cells and then looked at different methylation patterns in the promoter regions. So one result that she found from this that I wanted to mention because it correlates with the results that I just spoke to you about was that she saw that DNA from the blood mononuclear cells that had been heat stressed and isolated from high and low immune responders did show different methylation patterns in the promoter region that indicated that the heat stress cells from the high immune responders may have increased expression of genes related to heat shock protein and genes involved in cellular protection and survival. So that corresponds with that heat shock protein 70 data that I just showed you. So now I'm going to move to talking about our next objective, which was looking at the effect of in vivo heat stress on physiological parameters and the function of blood mononuclear cells in immune phenotype dairy cattle. And this study is currently under review in veterinary immunology and immunopathology. So our hypothesis for this study was that cattle classified as high immune responders will have lower respiration rates and rectal temperature, as well as greater heat shock protein 70 concentration and cell proliferation compared to average and low. So for this study, we used a subset of 8 high, 8 average, and 8 low lactating cows that I had mentioned previously in the first objective. And then we housed these cows in groups of 6. So each group had 2 high, 2 average, and 2 low. And they were housed in the metabolic wing of the Elora Research Station. And then we exposed these cows to a high THI for 4 hours on 2 subsequent days. So the idea here was that we were hoping to mimic that same heat challenge that we just did on the cells in vitro, but now exposing the cows to that heat challenge. So we turned the cooling fans off in this area, as well as turned the temperature up in the tie stall wing to try and create that heat challenge environment. And then we took blood samples from the cows, both pre and post the heat treatments on each day, as well as looked at respiration rate and rectal temperature every 30 minutes during the heat treatments and pre-heat treatment as well. And then we isolated blood mononuclear cells from the blood to evaluate cell proliferation and heat shock protein 70 concentration. And then used repeated measures to count for any effects in respiration rate and rectal temperature, and general linear models to account for differences for cell proliferation and heat shock protein 70. So looking at the results for this study, this first graph is showing our results for respiration rate in breaths per minute. So as you can see from this graph, as we get out to the higher THI values, we did find a significant difference between our phenotypes, with our highs having significantly lower respiration rate compared to our average and lows. And another thing I wanted to point out was that as we get to a THI of 76 to 77, this is where we're seeing our average and lows start to have respiration rates above normal, which is indicated by this black line here, whereas we don't see our highs having respiration rates above normal until we get out to a THI of 78 to 79. And then this next graph is showing our results for rectal temperature. So we didn't see any significant differences between the phenotypes for rectal temperature. And in general, the cows in the study remained within normal ranges for rectal temperature across the different THI values. However, we did notice that at THI of 76, we do see a significant increase in rectal temperature compared to our lower THI values. And this rectal temperature remained high for the duration of the other THI values. So this increase corresponds with what we saw for our average and lows for respiration rate. And then this graph is showing our results for HSP 70 concentration. So from this graph, you can see that after our first heat stress treatment on the cows, the highs had significantly greater HSP 70 production compared to both our average and lows. Additionally, after the 2 subsequent heat treatments, we also saw that they had a trend to have greater HSP 70 production compared to average and lows. And then when we look at within phenotype across the treatments, we found that the highs had a significant increase in HSP70 production compared to thermoneutral after both of our heat treatments. And then after the heat treatments at our 18-hour measurements, we do see a decrease. And this is typically, this pattern is what you would expect to happen for HSP70 production. So when you have that that threat of a heat stress, you'll see HSP70 increasing, but once that threat is no longer a threat, then you actually do see a decline in HSP70 production. And then for our averages and lows, we actually didn't see any significant difference in HSP70 concentration across our different treatments. And then this graph is showing our results for cell proliferation. So again, we're seeing after our 2 heat stress treatments, after the first heat stress treatment, we see a significant increase in cell proliferation compared to our lows. And then additionally, after the second heat stress treatment, we see a trend towards greater cell proliferation compared to averages, and again, a significant increase in cell proliferation compared to the lows. And then looking across or within phenotype across the treatments, again, we're seeing that significant difference or a trend towards a significant difference between the thermoneutral for our heat stress 1 and heat stress 2 treatments in the highs, but then see no significant difference in cell proliferation across the treatments for our average and lows. And studies have also shown that cell proliferation and HSP70 production are actually linked. So these results do correspond with what we were seeing for our HSP70 production. So I also just wanted to compare the results from our first objective to our second objectives. So this first graph is showing our results again that we found in the in vitro heat challenge on the cells versus what we saw in vivo when we did the heat challenge on the cows. And from these 2 graphs, we are seeing similar results in the fact that our high responders tend to have greater heat shock protein concentration after our 2 heat treatments. And then additionally, when we compare our results for cell proliferation, again, we're seeing similar results in the fact that our high responders have greater cell proliferation compared to our average and lows after the 2 heat treatments. So this might indicate that our in vivo studies are representative of what's happening in vivo. So then moving on to our next objective, we looked at the physiological responses to heat stress in immune phenotype dairy cattle in freestall and high stall management systems, which this study is actually just found out today is now under review in Frontiers in Animal Science. And the hypothesis for this was that cattle classified as high immune responders will have lower respiration rates and rectal temperature in both freestall and high stall management systems. For the sake of time for today, though, I'm only going to present to you what we saw in the high stall management system, just so we can compare it back to that previous objective that I just talked about. So for this study, we used a total of 6 high and 6 low lactating cows that we split into 2 groups of 3. And they were housed again in the tie stall area at the Allura Research Station. So for this study, we didn't turn off any of the cooling fans, and we just left it as is to expose them to more of a natural heat challenge versus the induced one that I was mentioning in the previous objective. And then we took manual rectal temperatures twice daily for a consecutive 12 days for each group. So in total with the 2 groups, it was over 24 days. And we did this at the end of July to the end of August. And then we also, the cattle were equipped with respiration bands to measure respiration rate. And then we also recorded corresponding temperature and humidity data in order to calculate temperature humidity index, and then used repeated measures to analyze this data. So this slide here is showing a picture of what the respiration bands look like that we had on the cattle. Okay, so now moving on to the results. This first graph is showing our results for respiration rate. So as you can see from this graph, our high immune responders, which is shown in the green here, had significantly lower respiration rate across a number of different THI values compared to our lows. Additionally, again, when we get out to that THI of 76, 77, we're seeing our low responders have an increased respiration rate above normal, versus we're not seeing our high responders have an increase in respiration rate above normal until we get out to about a THI of 80. And then when looking at results for rectal temperature, again, we didn't see a significant difference between the phenotypes for rectal temperature. And in general, most of the cattle did remain within normal ranges across the study. But one thing I did want to point out, again, when we look at that THI of 76, that seems to be, at least when our low cows, again, are having that increase in rectal temperature relative to the lower THI values. So then comparing our results from that previous objective for the physiological mechanisms, these first graphs showing what we found for respiration rate. Again, we're seeing a significant difference in the phenotypes with the high immune responders having lower respiration rates as we get out to the higher THI values. And similarly, in the two studies we saw for our lows or our average and lows, the respiration rate going above normal in that THI range of 76 to 77. Versus our highs not having respiration rates above normal until we're getting out to THIs of between 78 to 80. And then for rectal temperature, again, in both studies, we didn't find significant differences between the phenotypes, but similarly in both studies, when at that THI of 76, is where we were seeing increases in rectal temperature above what we were seeing in our lower THI values. So seeing similar results in the physiological measurements between those two studies that we did. So now I'm going to move on to our fourth objective and the last objective I'm going to talk to you about. So because we were the studies previously have indicated that there might be a difference between the immune phenotypes and their ability to respond to heat stress. We wanted to know why this might be the case. And one idea that we had is maybe it was due to differences in their cellular metabolism. And actually some of the results that Ali found in her paper too did point to the fact that there could actually be differences in cellular metabolism between our high and low phenotypes. So the objective for this study was to look at the effective in vitro heat stress challenge on cellular metabolism of blood mononuclear cells from immune phenotype dairy cattle, with our hypothesis being that the cellular metabolism pathways used by blood mononuclear cells to generate energy at thermoneutral and during heat stress would differ between high and low immune responders. So for this study, we collected whole blood thermoneutral THI from 10 high and 10 low lactating cows at Elora Dairy. And then we isolated blood mononuclear cells from these cows and then exposed them to the 3 heat treatments that I talked to you about previously in that first objective. And then we looked at different cellular metabolism outputs. So we looked at glucose and lactate as an indicator of glycolysis. as well as acetyl coenzyme A as an indicator of fatty acid oxidation, and it's also an input for ketogenesis. And we did this using commercial kits. Additionally, we also looked at these 10 high and 10 low cows, but collected sera from them at thermoneutral and heat stress times to assess beta hydroxybutyrate and fatty acid concentration. So these we didn't look at in the blood mononuclear cells because typically, well, for beta hydroxybutyrate, it's produced in the liver and then goes into circulation. And then the immune cells will take it up from circulation. And additionally, for fatty acid concentration, it's also taken up from circulation to be used in the fatty acid oxidation cycle. And I also, just to give you a little background about why we're looking at these different metabolites. So typically during a non-stressful event or possibly at thermoneutral conditions, immune cells will use the oxidative phosphorylation cycle in order to produce energy. However, during possibly heat stress or a stressful event like disease, there's no longer enough oxygen available to use the oxidative phosphorylation cycle in order to produce energy. So then studies have shown that typically immune cells will switch more to the glycolysis cycle in order to produce the energy needed during those stressful events. Okay, so then to analyze this data, we used general linear models. So now I'm going to move on to talking about the results from this study. So this first few graphs is showing our results for glucose concentration. So this first graph showing between the phenotypes within each heat treatment. So at thermoneutral, we found that our low responders had a trend towards greater glucose production compared to our high immune responders. So possibly indicating already at thermoneutral that those low responders are using the glycolysis cycle versus oxidative phosphorylation. Versus when we get out to after 2 heat stress treatments, we then see a switch to our high immune responders producing more glucose concentration compared to our lows, possibly indicating that now the highs are switching to using more of that glycolysis pathway versus our low immune responders. And then when we look at within phenotype across the treatments, we do see a significant increase in glucose concentration compared to thermoneutral for the highs after our 2 heat stress treatments versus the lows. We didn't see any significant difference across the treatments for our glucose concentration. However, did still see at thermoneutral that it was higher relative to after heat stress. And then when we look at our results for lactate concentration, which is that output of the glycolysis cycle, we see a significant increase at thermoneutral for the lows compared to highs for lactate concentration. But then after our 2 heat stress treatments, we're actually seeing that switch again with our highs having a trend towards greater lactate concentration compared to our lows. So the patterns that we saw for glucose are quite similar to what we're seeing for lactate. So might indicate that at that thermoneutral, our lows are using the glycolysis cycle more, but then after heat stress, we see our highs using it versus our lows. And then looking at results within phenotype across the treatments, we see that at heat stress, our highs do have a significant increase in lactate production compared to at thermoneutral, but then we do see a significant decrease from heat stress 1 to heat stress 2. And then in the lows, we're seeing from thermoneutral to heat stress 2 a significant decrease in lactate concentration. And then this next slide is showing our results for fatty acid concentrations, so the input for the fatty acid oxidation cycle. So from this first graph, we can see that actually at both thermoneutral and heat stress, our lows have significantly greater fatty acid oxidation concentration compared to our highs. So possibly indicating that they're using this cycle both at thermoneutral and heat stress more than what our highs would be. When we look at within phenotype, though, across the treatments, we do see a significant increase in fatty acid oxidation concentration compared to at thermoneutral for our highs. So could indicate that the highs are using fatty acid oxidation more at heat stress more than what they would be at thermoneutral. But again, when we go back to comparing between phenotype, it seems that our lows tend to use it more readily than our highs would be. And additionally, we see an increase in fatty acid concentration at heat stress compared to thermoneutral for our lows as well. And then this graph results for coenzyme A concentration, which is the output of the fatty acid oxidation cycle. So again, we're seeing our lows having significantly greater coenzyme A concentration across our 3 heat treatments compared to our highs. So that's having that same similar pattern as what we saw for fatty acid concentration. And then when we look at within phenotype across the treatments, we saw that there was a trend towards our highs to have a significant decrease in coenzyme A concentration from heat stress 1 to heat stress 2, whereas we didn't see any significant difference for the lows across the treatments. And then this slide is showing our results for beta hydroxybutyrate concentration. So we didn't see a significant difference between the phenotypes in the thermoneutral or heat stress treatments. And additionally, when looking within treatment, within phenotype, we also didn't see a significant difference between thermoneutral and heat stress for the highs or the lows. So for this study, we made sure that the cattle were lactating as well as were far enough away from calving that they shouldn't have been in a negative energy balance, so that we were just looking at the effect of heat stress itself on beta hydroxybutyrate concentration and finding that there doesn't seem to be an effect, and this actually does correspond with what has been shown in some other studies. So in conclusion, our results suggest that high immune responders may be tolerant of heat stress caused by climate change compared to average and low responders, and this is due to their ability to produce greater concentrations of heat shock protein 70 and nitric oxide, as well as have greater cell proliferation when exposed to heat challenges. We also saw in a couple of our studies that they tend to have lower respiration rates at higher THI values. Additionally, our results were similar for HSP70 concentration and cell proliferation between our in vitro and in vivo studies, which may indicate that our in vitro experiments provide a good indicator of what is happening in vivo. Additionally, our results suggest that at thermoneutral, low immune responders tend to use the glycolytic pathway more than the high immune responders to produce energy, whereas during heat stress, the high immune responders tend to use the glycolytic pathway more compared to lows. Additionally, we saw that at thermoneutral and heat stress, lows also tend to use fatty acid oxidation pathway more than the highs to produce energy, which suggests differences in cellular metabolism between our immune response phenotypes, and may be one reason why we see differences in response to heat stress. So thank you very much for your attention. I'd just like to also thank my advisor, Dr. Bonnie Millard, and my committee members for all their help and support with this, as well as my various lab members and our different funding agencies. So I'd be happy to take any questions you might have.
Thank you very much, Shannon. I'm going to now change the meeting. So if you have any question to Shannon and you want to ask yourself, you can now unmute yourself and have your camera on if that's the case, or you can also chat in the chat box and I will read the question to Shannon.
Okay. Can you still see the slides, just like out of presentation view?
Yes, the PowerPoint.
Okay. I just switched because I couldn't see you guys anymore. I kind of lost that after I lost connection. So
Okay.
I just switched it so that I can now see the camera as well. I just want to make sure you can still see the slide.
All right, so I think I saw a hand. I see Flavio. So Flavio, if you want to go ahead.
Okay, can you hear me, Shannon?
Yeah, I can hear you.
Okay. I have a question for you. In your experimental design, when you have the groups of high, low, and the average group of cows, how did you account for the possible sire effect?
Possible sire effect?
Yes.
We didn't actually account for sire effect in general. We were just basing it. So we did like these, the high, low, and average are calculated based on breeding values. But the idea was we wanted animals that were high, high, average, average, and low, low for each of the phenotypes. So that really limited the animals that we could use. So we basically, our selection was based on the fact that they were high and low for the, based on their breeding values for antibody and cell-mediated immune response.
But have you looked at the groups, if by chance given, let's say a high group was all daughters from the same sire?
I haven't looked at that, no.
Or the distribution, or you know what I mean?
Yeah, no, I do know what you mean. Yeah, no, I haven't looked at that, but yeah, that's definitely something that we could look into to see, to make sure that it isn't, like a sire effect versus something else. Yeah.
Yeah, maybe something to think about when if it's possible to get groups that will have a balanced contribution, or not coming at least like all high, example, from one single side, all lows from one single side, right?
No, I know what you mean. Yeah. I should definitely look into that to make sure that they aren't all coming from the same sire, but yeah, I say we were limited on who we could choose, so we were basing it on phenotype versus looking into those. But yeah, I should make sure that it isn't a sire effect versus the phenotypic.
Another second question is, I noticed that in the first experiment, you set as 42 degrees for the heat stress of the cells, right?
42, yeah.
Yeah, but in the fourth one, you decreased to 41 degrees. Why was that?
So that the main reason for that is we were doing these in incubators, and we have our own incubator, and then we're sharing an incubator with the other lab that is the one that sets the higher temperature. So when I did the first study, they had that set to 42 degrees, but then when I went to do the next one, they had it set to 41. And I can't, because it's not my incubator, I can't adjust the temperature to what I want it to be. So yeah, we did have that 1 degree difference, but in the grand scheme, I think it necessarily made a huge impact to the heat challenge. But yeah, that was a little bit unfortunate that
Yeah, was not based on the results from the first study and compared with the in vivo that you decide to lower the temperature. No?
No, it was
Yeah,
just a function of, yeah.
Why I ask you this, Shannon, because I always wonder when you are doing in vivo and let's say you're applying a heat stress of 42 degrees, how much of the heat stress in vivo you have to apply to have a corresponding result, right? Because we know, because for a cow to have a fever of 42 degrees, the heat stress, the cow is going to die. You know what I mean?
Yeah, it would be pretty hot. So we did actually that.
So
Sorry.
that's why I thought you decreased it in the second, the fourth experiment, because 42 is really high, right?
Yeah, it is high, but it does actually, there are a lot of studies that when they've done in vivo work on cattle that it's similar, they are using that 42 degrees. So I mean, it was partially what we had available, but it also is backed up by the literature. But yeah, I do understand what you're saying. If they were exposed to 42 degree, like if they had a body temperature of 42 degrees for 4 hours, that yeah, they probably wouldn't be doing so well.
Okay, thanks, Shannon. That's what.
No.
All right, so Shannon, we got a question in the chat. I don't know if you can see it.
I did see it pop.
If you can, I can read it.
No, I saw it pop up, but I can't see it now.
That's okay. So Jason is asking, do you believe it would be feasible to create GEBVs for immune response?
So that actually is kind of already being done. So I don't know if anyone is aware of the Immunity Plus line of sires with CMEX, but they actually, when they denote sire as Immunity Plus, they are actually basing it on GBVs. So in the introduction, when Ivan was mentioning that I worked on that project that was looking at creating a genomics test for immune response, project was actually successful and we were able to create a genomics test, but it was licensed to CMEX, so it's exclusively being used by them at this point. So hopefully that answers his question.
Yeah.
Yeah.
All right, Shannon, I have a couple of questions. As you know, I'm also working with heat stress and using the temperature humidity index. So my question is, just to start, I was wondering, how did you calculate the THI? Was it using the formula you showed us in the presentation?
Yeah, it was. So I know you've used a different formula, correct?
I actually used the same.
Oh, did you? Okay.
Yes, I worked actually with two ways of calculating the THI. The formula is the same. The difference is the parameter that you include in the formula. So in one is the maximum temperature, the daily maximum temperature, and daily minimum relative humidity. But the other one that I also calculated is just the daily average temperature and relative humidity. It was that one that you worked with, the average, daily average?
So because like I had specific time points that I was looking at, I actually used the temperature and humidity for those specific time points.
Okay.
Do you know what I mean? So it wasn't an average. So like when I because obviously my data does not span as much as your data. Like, so because I'm just looking at like a snapshot in time, and then like for that second objective, I was just looking at every 30 minutes during that 4-hour period, then I would use the temperature and humidity at that time that I took the rectal temperature or respiration rate temperature.
Yeah, I was wondering because to me the values of DHI, they seem to be a little higher than what I was expecting.
Oh, okay.
Yeah.
So it was also, I didn't show the data from the freestall area. So my THI values from my freestall area are actually a lot lower than in the tie stall area. So those out in the freestall area, I only ever reached, I believe, a THI of 78, maybe 79. I didn't get to 80. So that might correspond more to some of the values you were seeing.
Yeah, and the values, the parameters they used, they were all collected from the inside the barn, right? In LoRa?
Yes. Yeah.
Okay.
Yeah. So, yeah. So anyway, the higher THI values are values that you're seeing from that tie stall area. I don't know if you've ever been in a LoRa or not.
Yes.
So if you know that tie stall area is not exposed to any like outside walls at all, and is only getting airflow basically from that tunnel ventilation from the outside. So it actually does get a lot hotter in there versus what you're getting in that freestall area. And then again, in that second objective, we actually turned the temperature up in that area, as well as turned our ventilation fans down and turned off all the cooling fans. So that's also why we likely got like higher THI values from what you would be used to seeing in like your data, I would assume.
Yeah. Yeah, no, that's interesting. Yeah. And speaking of the tie stall and freestall, sort of, you mentioned there is a study. You showed the tie stall, right?
Yeah.
And the hypothesis was considering high immune response regardless the stall. It's either tie stall or freestyle. But I was wondering if you have sort of something to elaborate more in the difference between these two, considering not just the high immune, but what would be the difference that you are aware of in terms of the mechanism, the physiological mechanisms to cope with heat?
Between the phenotypes, you mean?
Yeah, it's well, basically the question is about the hypothesis considering two different housings. So what is the assumptions considering the mechanisms that are they different in between these two, or yeah?
No, I don't. I think the mechanisms would be similar. We did actually find again, even though we didn't reach that higher THI values in the freestall, but when we did get out to like the 77, 78, our high immune responders still did have significantly lower respiration rates. So we were seeing similar results in both of those. But we did find when we compared the THI values to the freestall compared to what we see in the tie stall, there is like the tie stall did get significantly hotter than the freestall. But I think in terms of like internal mechanisms that each phenotype's using, I think it doesn't really, like they're probably using similar ones in both housing systems. I think maybe just our high immune responders, they just might deal with stressful situations in general better, just because we've shown that stuff with disease before, and now we seem to be seeing similar results with heat stress as well. I don't know, does that answer your question?
Yes. Yeah, thank you, Shannon. Yeah,
Okay.
that was actually my bad in terms of formulating the question, but that was the idea. Thank you for your answer.
Yeah, I could have shown that freestyle. So I just, for time wise, I didn't think I had enough time to show everything. So, which is why I just chose to pick the ones that I could compare to some of our previous objectives. So, yeah.
That's all right. We received another question from Murilo, so I'm going to read for you. Thanks for the great presentation. Have you looked at performance, so fat yield and fertility, on the low versus high animals?
No, we haven't looked at fat yields. We have included production parameters in some of our models, and we didn't see any significant difference in production. But I think, as you'll see, a lot of studies show you don't start getting that decline in production until you have actually differences in body temperature as well. So I think we weren't able to induce enough of a heat stress to start seeing those declines in production, which is why we didn't have any differences. But in terms of looking at fat yield, no, I have not looked at that. Specific reproduction parameters, also no. I do have another study where I did look at prolactin concentration that I didn't present here for sake of time. But so in that, we actually didn't see any differences in the prolactin concentration either. But I also think, again, that study was done in the maternity wing, so kind of similar to the freestall area. And we didn't get out to a THI much past, I believe, 78 again, if I'm remembering correctly. So I don't think we were able to induce enough of a heat stress to actually make like see any differences between phenotypes, as well as differences between like the thermoneutral and heat stress treatment. So yeah, I guess that was kind of a long-winded question or answer to her question, but yeah, no, we haven't looked specifically at any of those parameters. We were initially thinking of doing that, but then we kind of went a different direction with the project.
All right, so does anyone have a final question to Shannon? Okay, if not, I think that was it. Thank you, Shannon, very much for a very good presentation and accepting our invitation to start us off.
Thanks for having me. Sorry I didn't have a whole lot of genetic stuff in there, but hopefully everyone still found it informative and interesting.
Yeah, I'm sure it was good.
Okay, thank you.
Yeah, and thank you all for attending today's seminar, and I hope to see you next week. Have a good afternoon.
You too. Thank you.